The Cogitating Ceviché presents Rosalind Franklin, Unraveling the DNA of the Digital Age. Past Forward, Historical Icons in the Digital Frontier No. 9. By Conrad Hannon. Narration by Amazon Polly. Preface. As we continue our intriguing exploration in the Past Forward, Historical Icons in the Digital Frontier series, this 9th installment delves into the life and legacy of Rosalind Franklin, whose profound contributions to the understanding of molecular structures have rippled through time to influence today's technological and scientific arenas. Each article in our series has reimagined the past's luminaries in today's digital world, illuminating the timeless nature of their discoveries and their speculative interactions with modern technology. In this edition, we honor Rosalind Franklin, whose meticulous work with X-ray crystallography provided the world with its first glimpses of DNA structure. This discovery has become the cornerstone of genetic research and biotechnology. Her dedication and precision in capturing the hidden details of molecular formations have paved the way for innovations that were once the realm of science fiction, from gene editing with CRISPR to digital data storage using DNA. As Past Forward explores the hypothetical scenario of Franklin in the modern world, we find her legacy intricately woven into the fabric of contemporary science. From her potential contributions to viral research amidst global health crises, to her hypothetical engagement with artificial intelligence in computational biology, Franklin's influence extends across disciplines, underscoring the importance of an interdisciplinary approach and ethical rigor in scientific exploration. Franklin's story is particularly poignant, not just for her scientific acumen, but for her resolve in the face of the numerous challenges she faced as a woman in a male-dominated field. Her journey, as imagined in this series, serves as an inspiration and a call to action, reminding us of the critical role of diversity and inclusion in driving scientific progress. Though speculative in these narratives, her voice resonates with urgency and authority, advocating for equity and recognition for all contributors in the scientific community. Join me as we navigate the fascinating landscape of Past Forward, Historical Icons in the Digital Frontier. This journey not only celebrates the achievements of those who laid the foundations for our understanding of the world, but also challenges us to think critically about the direction of future innovations and the ethical dimensions of our scientific endeavors. Let us continue to draw inspiration from Rosalind Franklin's legacy as we forge ahead, exploring new frontiers in science and technology with integrity, curiosity, and respect for the past that shapes our present and molds our future. Onward, as we venture further into the digital age, guided by the spirit of a visionary whose work continues to enlighten and inspire the path of scientific discovery and technological innovation. Conrad T. Hannon. Introduction. A Visionary Reimagined. Imagine Rosalind Franklin stepping into the 21st century, her eyes widening as she beholds the landscape of modern science, a realm transformed by the very molecular structures she once illuminated under her X-ray crystallography experiments. The world she encounters is one where genetic engineering is not just a theoretical concept, but a tangible practice that shapes life at its core. Here, the DNA double helix, whose structure she crucially contributed to revealing, has become the foundation for breakthroughs in biotechnology and medicine. As Franklin navigates this new world, her initial astonishment transitions into a profound engagement with the technologies her work has helped foster. She is introduced to CRISPR-Cas9, a gene editing tool that performs with precision she could only have dreamed of, directly manipulating the genetic fabric of living organisms to cure genetic diseases and improve agricultural crops. Observing these applications, she marvels at how the seeds of her research have blossomed into tools that not only understand, but also actively reshape the blueprint of life. The tone of wonder mixed with a rigorous scientific curiosity sets the stage for Franklin's exploration of this advanced era. As she interacts with contemporary scientists and technologists, her sharp insights and steadfast dedication to empirical evidence drive her to delve deeper into the ethical, social, and scientific implications of these developments. Franklin's journey through the modern landscape is not just a testament to her enduring legacy, but also a dynamic engagement with the possibilities and responsibilities that come with the power to decipher and redesign the very essence of life itself. Genetic engineering and biotechnology. Rosalind Franklin stands in a state-of-the-art genetic engineering lab, observing a demonstration of CRISPR technology with a mix of fascination and critical thought. Her pioneering work in capturing the first clear X-ray images of DNA structure now echoes in this room, where scientists manipulate genes with unimaginable ease in her time. She watches as they edit genetic sequences precisely, correcting mutations that cause disease and enhancing crop resilience to climate change. Franklin's reaction to CRISPR and similar technologies is multifaceted. On one hand, she is captivated by the power and potential of these tools, which directly apply the foundational knowledge of molecular structures she helped uncover. On the other hand, she remains acutely aware of the profound responsibilities that accompany such power. Her methodical and meticulous approach to science makes her cautious about the rapid application of gene editing technologies without a thorough understanding and consideration of long-term effects. In discussions on the ethical implications of genome editing, Franklin becomes an advocate for rigorous scientific integrity and responsibility. She urges the scientific community to implement robust ethical frameworks that ensure these powerful technologies are used to benefit humanity without compromising ecological or individual integrity. Franklin also addresses the potential risks of gene editing, such as unintended consequences in genetic interventions and the ethical dilemmas posed by enhancements and non-therapeutic modifications. Moreover, Franklin takes a keen interest in the role of women in science and technology, a topic deeply personal to her given her experiences of gender bias during her career. She is both heartened by the progress made in gender equality and motivated to push for greater inclusion and support for women in STEM fields. Franklin engages in mentorship and advocacy, using her stature to encourage young women to pursue careers in science and technology, ensuring they have the opportunities and recognition often denied to her and her contemporaries. Through her engagement with genetic engineering and biotechnology, Franklin revisits her legacy in the light of modern science and contributes to shaping its future. Her voice becomes crucial in guiding the conversation on how these technologies should evolve, balancing scientific advancement with ethical considerations and striving for a more inclusive scientific community. Digital data storage, from DNA to data. Upon discovering that DNA, the molecule whose structure she had famously helped to elucidate, is now being used to store digital data, Rosalind Franklin's intellectual curiosity is immediately piqued. The concept of encoding vast amounts of information into the compact, helical strands of DNA represents a fascinating convergence of her past work with the latest advancements in technology. She is intrigued by the elegance of using DNA's natural storage capabilities to address modern challenges related to data density and retrieval. Franklin marvels at the efficiency of DNA as a storage medium, capable of housing an exabyte of data within a gram of material, with durability that could withstand thousands of years if properly preserved. She appreciates the irony of this biological system, which she studied to understand life's blueprint, now engineered to safeguard human knowledge from ancient texts to contemporary archives. Her expertise in X-ray crystallography, which allowed her to see the underlying structure of DNA, offers metaphorical insights into solving problems of data compression and encryption. Franklin contemplates how her method of discerning fine details in molecular patterns could be adapted to improve the fidelity and efficiency of encoding and retrieving digital data from DNA. She proposes using similar crystallographic principles to refine the methods by which data is translated into genetic sequences and back, ensuring accuracy and minimizing errors. Additionally, Franklin is drawn to the challenges of scalability and accessibility in DNA data storage. She engages with engineers and biotechnologists to explore innovative ways to streamline the process, making it more viable for widespread use. Her approach is methodical and grounded in empirical research, advocating for thorough testing and validation to ensure that DNA data storage technology can be practical and cost-effective. Throughout her exploration of this cutting-edge field, Franklin remains deeply involved in the scientific discourse, contributing her unique perspective on the intersection of biology and technology. Her legacy in X-ray crystallography becomes a touchstone for developing new techniques in the digital realm, inspiring a cross-disciplinary approach that seeks to leverage the best of both worlds, the stability and compactness of biological systems with the dynamism and scalability of digital technology. Viral research and public health. Rosalind Franklin's hypothetical foray into the realm of modern viral research, especially amidst global challenges such as the COVID-19 pandemic, opens a new chapter in her scientific journey. Known for her meticulous work with X-ray diffraction techniques, Franklin would be captivated by the sophisticated imaging technologies now used to study viruses at the molecular level. Tools such as cryo-electron microscopy, which have evolved from principles she pioneered, are instrumental in visualizing viral structures and have been crucial in the rapid development of vaccines. Franklin's methodological approach and keen analytical skills would naturally lead her to contribute significantly to understanding how viruses interact with human cells, the mechanisms by which they invade and replicate, and how these processes can be inhibited. She would be deeply involved in the structural biology of viruses, applying her expertise to map out the proteins on the surface of viruses that are key targets for vaccines and antiviral drugs. Given her interdisciplinary background and known propensity for collaboration, Franklin would likely drive initiatives to foster cooperation between biologists, chemists, and physicists to integrate different scientific perspectives to fight against viral diseases. She advocates using advanced imaging techniques to understand the basic biology of viruses and to guide the design of vaccines and therapeutics. In the context of vaccine development, Franklin's contributions would be particularly valuable. She would understand the importance of structural insights into viral proteins in designing vaccines that elicit a robust immune response. Her work could lead to breakthroughs in vaccine efficacy and delivery, particularly for RNA-based vaccines, which hinge on precise molecular interactions that her imaging techniques could elucidate. Moreover, Franklin's commitment to public health would extend beyond the laboratory. She would be vocal about the need for equitable access to healthcare and the global distribution of vaccines, especially in underserved regions. Her advocacy would stem from a deep understanding of the global interconnectedness highlighted by the pandemic and the role of science in bridging health disparities. Franklin's involvement in modern viral research would not only honor her legacy in molecular biology, but also underscore the continuing relevance of her scientific approach in addressing some of the most pressing public health challenges of our time. Her theoretical contributions to viral research and vaccine development would blend seamlessly with her enduring impact on the scientific community, providing a multidimensional perspective that enriches science and its application to human health, AI, and computational biology. In the high-tech corridors of computational biology, Rosalind Franklin would find herself at the confluence of her expertise in X-ray crystallography and the burgeoning field of artificial intelligence. Her fascination with the molecular structures of DNA and viruses would naturally extend to AI's capability to model complex biological processes and structures, providing insights that would otherwise take years of meticulous laboratory work. Franklin would be particularly intrigued by how AI algorithms predict the three-dimensional structures of proteins and nucleic acids. These tasks echo the challenges she faced in her crystallographic endeavors. She would see the potential of machine learning techniques to transform her static images into dynamic models that show how molecular structures interact and change over time, bringing a new depth to her understanding of life's building blocks. Her methodical nature would drive her to work closely with computer scientists and bioinformaticians to refine these AI models. Franklin would advocate for integrating high-quality empirical data, akin to her precise crystallographic data, into training datasets for machine learning algorithms. Her goal would be to enhance these tools' accuracy and predictive power, ensuring they reflect the true complexity of molecular biology. Moreover, Franklin's keen scientific intuition would lead her to explore the limits and biases of computational models. She would develop new algorithms that could better account for the irregularities and unique characteristics of different molecules, potentially leading to more personalized approaches in medicine and pharmacology. Her contributions would push the boundaries of computational biology and ensure that these technological advancements are grounded in robust scientific principles. Additionally, Franklin would likely be involved in projects using AI to sift through vast datasets derived from global biobanks, looking for patterns and correlations that could lead to new discoveries in genetics and epidemiology. Her analytical skills would be invaluable in deciphering complex data, identifying significant biological markers, and proposing novel hypotheses about health and disease. In this digital era, Franklin's hypothetical engagement with AI and computational biology would represent a natural evolution of her work. By merging her deep understanding of molecular structures with advanced computational methods, she would help to usher in a new era of scientific discovery, where artificial intelligence becomes a pivotal tool in the quest to decode the complexities of life. Her legacy, deeply rooted in the precision of crystallography, would thus expand to influence the cutting-edge fields of AI-driven research, continuing to inspire a generation of scientists to look deeper into the fabric of the natural world. Conclusion: a legacy beyond the helix. As we encapsulate Rosalind Franklin's journey through the modern digital and scientific landscapes, it becomes evident that her influence extends far beyond her seminal contributions to the structure of DNA. Franklin's work, marked by an indomitable spirit of inquiry and exacting standards, resonates deeply within today's multifaceted scientific endeavors, from genetic engineering to artificial intelligence and biology. Franklin's legacy is not merely preserved in the helical structures she helped uncover, but vibrantly alive in the ongoing advancements that her pioneering techniques have made possible. Her approach to scientific research, rigorous, detailed, and profoundly analytical, has set a standard that continues to inspire a new generation of scientists and researchers. The meticulous methods she employed in X-ray crystallography have found new applications in areas she could hardly have imagined, influencing not just molecular biology, but also computational modeling and data analysis across various scientific disciplines. Moreover, Franklin's interdisciplinary approach, which seamlessly blended physics, chemistry, and biology, underscores the importance of crossing traditional boundaries to foster innovation. In an era where the lines between disciplines increasingly blur, her work exemplifies how integrating diverse scientific perspectives can lead to breakthroughs that transcend the sum of their parts. Today's researchers draw on her model to tackle complex problems, from understanding the mechanisms of life at a molecular level to developing technologies that safeguard our health and environment. Her unwavering commitment to empirical evidence and ethical considerations remains particularly relevant in today's rapidly evolving technological landscape. As scientists and technologists push the boundaries of what is possible, Franklin's legacy serves as a reminder of the need to ground advancements in solid research and consider their broader impacts. Her life's work encourages ongoing dialogue about the ethical implications of scientific research, ensuring that technological progress enhances societal well-being. Reflecting on Rosalind Franklin's impact, it is clear that her legacy is not confined to the past but continues to influence the future. Through her eyes, we are reminded of the enduring power of curiosity and meticulous research, inspiring us to pursue knowledge with both passion and integrity. Franklin's journey through the digital age, though hypothetical, offers a narrative of inspiration, urging us to embrace the complexities of science and technology with the same courage and thoroughness that she exemplified throughout her career. Her legacy teaches us that at the intersection of various scientific fields lies the potential for revolutionary discoveries, and it is here that we must continue to explore, innovate, and above all, strive for excellence. Thank you for stopping by, and until next time, stay gruntled.